Good afternoon, everyone. Welcome to our MHRC seminar this afternoon. My name is David Hood. It's a pleasure for me to introduce our speaker today, Dr. Bernadette Murphy. Dr. Murphy is in the Faculty of Health Sciences at Ontario Technical University in Oshawa. She has a very interesting background. She did her undergraduate degree at Queen's University in Life Science. Then she went to the Canadian Memorial Chiropractic College and got a degree in chiropractic medicine. Then went to Auckland, New Zealand, where she did her master's degree and her PhD in the exercise science program, and spent 10 years in Auckland, New Zealand before returning to Canada, where she is now at the Ontario Tech University. And she began the kinesiology program in that institution. And has been there for about 15 years, supervising graduate students in an NSERC funded program. So we're very excited to hear about your work, Bernadette, today. And the title of her talk is Effective Muscle Fatigue on Sensory Motor Integration. So Bernadette, please share your screen with us. And I should say also that Bernadette was selected by the MHRC Student Committee. So that's quite an honor. And Bernadette, you can start anytime you like. I can see your screen very well.
Perfect.
And, please go ahead and proceed.
Okay, well, thank you very much for inviting me, to Daniel and the Student Committee, and to David for introducing me. And I somehow managed to leave the word neck out of the muscle fatigue when I set the title. It must have been one of those days, because that is the focus of my research, and I'm going to explain why. Let's see here. Okay, there we go. So as I'm sure all of you are aware, particularly in this day and age, COVID has made it even more marked. So many of the activities that we do, whether it's occupational activities, recreational activities, the classic student posture, just activities of daily living, the neck is in a condition where it's under prolonged stress, but also often in a position for a long period of time that could result in neck muscle fatigue. And I'm a neuroscientist by training, and so I am interested in what are the neural consequences upstream when you have that sort of ongoing fatigue state. So just a reminder that neural plasticity is a change in the structure, function, or organization of neurons or nerve cells when they're exposed to new experiences. And normally we expect that or hope that that plasticity is what we call adaptive. It leads to somebody becoming an elite athlete or a virtuoso musician. But there's other times when that plasticity is maladaptive. And that's where you end up with people with chronic pain, chronic back pain, or carpal tunnel, or lateral epicondylitis. And just to give you an idea, I'm not going to go into details of this study, but this is a researcher named Herta Flor in Germany, just looking at fMRI activation and differences in response to sensory stimuli between a healthy brain and somebody who's got chronic pain. So what role might fatigue play in that? And I'll talk more about that soon. An important concept in the talk today is sensory motor integration. So a great example of sensory motor integration not working is when you reach and miss, but what it refers to is the ability of the central nervous system to integrate sensory information from different body parts and formulate appropriate outputs to muscles. The problem is that if you have maladaptive sensory input over time, that can also lead to maladaptive motor output. And then you can get into a loop where one is perpetually influencing the other. So the focus of my NSERC program really is based on the idea that you have altered input from the neck, whether it's by fatigue, postural stress, or going back to the chiropractic background, joint dysfunction, that is going to alter your body schema or internal body map, which then alters the way you process upper limb sensory motor integration. That's going to impact sensory motor integration, but also ultimately motor control, the ability to filter the sorts of sensory input that we generate when we do repetitive activity. It'll influence the ability to learn and perform motor skills. I'm not going to talk about this. This is just a possible application of it down the road. Could all of this gone wrong ultimately lead to chronic health conditions. But I'm focusing on the sort of upper part today because that's the focus of my basic science work. So I'm going to talk about 4 studies. And one thing I realized after, I wasn't planning to do all of Mabube's studies. So Mabube Zabihosenian is a student who did both her master's and PhD with me. And I'm going to talk about 2 of her master's and 2 of her doctoral studies because I think, or I thought it would make a nice story for your group because I know that people have a background sort of more muscle physiology, biomechanics. And so the first 2 studies sort of have a bit more of the biomechanics, and then I bridge into the hardcore neuroscience. So you can just, hold on to your hats when we hit the neuroscience. I'll try and go gently. Okay, so the first study that I'm going to talk about is neck muscle fatigue alters the cervical flexion relaxation ratio in subclinical neck pain patients. So subclinical neck pain is really recurrent neck pain, but why it's useful is it provides an opportunity to study chronic alterations in neck sensory input, but there's days when people are not in pain, so the way they move isn't altered, so we can look at some of the brain changes and those ongoing maladaptive changes. So people who have subclinical neck pain, they have decreased neck range of motion, decreased cervicocephalic kinesthesia, so less awareness of where their head is in space, and decreased muscular endurance. And as I mentioned, we can test participants on pain-free days and get an idea of those changes in sensorimotor integration independent of biomechanical changes in movement patterns. So the flexion relaxation ratio, it's a reliable method that can distinguish between healthy people and those with chronic or recurrent neck pain. And we know that the ratio is increased in neck pain patients because they cannot fully relax. Oh, I didn't define this. It's CEM is your cervical extensor muscles, and I will refer to that a few times in this talk. And fatigue is known to modulate both your flexion relaxation ratio, the ratio itself, as well as onset and offset angles in the neck. Okay, but nobody's really compared healthy and neck pain and how they respond to neck fatigue. So as I don't have to tell this group, I'm sure, but muscle fatigue has 2 components. It can both peripheral, which is the peripheral changes in muscle, and central, which is the decreased neural drive to motor neurons. Now, what we know is that when you have fatigue, it's going to affect sensory receptors. It's going to affect muscle spindle firing. And it's specifically by changing muscle spindle discharge rates, which decrease, and motor unit firing rates. And we also know that neck muscle fatigue reduces spinal stability. So the purpose of this first study was to determine whether performing a submaximal, so at 70% of maximal voluntary contraction, targeting the cervical extensor muscles would alter the cervical flexion relaxation ratio in both subclinical neck pain patients and a healthy control group. So the usual data acquisition, we used Trigno EMG to record over the cervical extensor muscles. And we use the Northern Digital's 3D investigator system. It's the poor cousin of the OptiTrack, but not too poor. It's still a 0.4 millimeter resolution to record the kinematics. And we had 13 healthy volunteers and 12 subclinical neck pain participants. So when we look here at the flexion relaxation ratio, fundamentally what happens is that participant starts in neutral and everything goes for 3 seconds. They then flex their neck, they hold for 3 seconds, then they re-extend their neck, and then they remain back in neutral. So we compared the average of 3 pre-fatigue trials, then we did our fatigue protocol, and then we did 3 post-fatigue trials for the ratio. So fatigue in this particular experiment was the participant was actually resisting against a force transducer. And what they did is they had to trace at the 70%. So the first we did 2 MVCs to establish what their maximum voluntary contractions were. Then they did the 70% ramp up and hold. So a 1 second ramp, 4 second hold. And they did it until they could not maintain that 70% force for 2 consecutive contractions. So to calculate the FRR, you're basically looking at the average EMG during the re-extension phase compared to relaxation phase. So the ratio will be lower if either you have increased activity in the so-called relaxation phase when your head is hanging fully in flexion, or if you have less, the ratio would also go down if you had less activity during the re-extension phase when the neck extensor muscles are doing their concentric phase of contraction. So this phase here is during neck flexion, the cervical extensors actually act as brakes. So this is the eccentric phase of the contraction. So we're comparing concentric to relaxation, and that's pretty standard in the literature. So the other thing we also looked at is the onset and offset angle of the change in the EMG activity to look if there was a change in the actual duration of that relaxation period. So the person assigning those was blinded as to which group people were in and whether it was pre or post fatigue, so that there was no bias introduced there. So when we looked at the fatigue, one interesting thing, just to start with, is that the healthy group had an average of 56 reps to fatigue and over 671 seconds, whereas the neck pain group had an average of only 39 and 473 seconds. So they did not have the same endurance. But when we look at the fatigability in terms of the mean power frequency and the root mean squared amplitude, both groups showed an increase in RMS and a decrease in mean frequency, significant decrease. So in both situations, we had EMG evidence that fatigue had indeed happened for both groups. When we looked at the flexion relaxation ratios, what we saw, first of all, as we expected at baseline, the dark is the baseline pre-fatigue. The neck pain group had a lower FRR, mainly because of that inability to relax in full flexion when your neck is just meant to be hanging on your ligaments, so to speak. But what was really interesting is after fatigue, the healthy control, their FRR went down. So almost looking a bit more like a neck pain participant. What was really interesting is that the neck pain patients actually went up a little bit in their FRR. So there was a significant group interaction, as well as that decrease in the FRR was significant for the control group. And then when we looked at the onset and offset angles, there was no group difference, but both groups after neck fatigue, what you saw was basically a change in both the onset and offset angles, so that you had a longer EMG silent period after fatigue. So we know that I mentioned earlier that the FRR can decrease due to either increased EMG and relaxation or decreased EMG during re-extension. So following fatigue, as we expected, the FRR was significantly higher in healthy compared to SCMP, consistent with past work. And that following fatigue, the controls decreased their FRR, but it was interesting that the neck pain group didn't. It may be because of the effort needed to generate adequate force. They had to have greater EMG activation in an attempt to just lift the head, which meant that the ratio actually increased instead of decreasing. Past work by others has shown that the neck muscles, as you might expect, they've got to hold the head up all day. They have more type 1 than type 2 fibers in healthy people. So it could be that a decrease in type 1 fatigue resistant fibers might be why the baseline differences in the neck pain group, because this has been shown to occur in the neck flexors in those with neck pain. That longer myoelectric silent period caused by earlier onset and later offset may be because the fatigued cervical extensor muscles can't stabilize the cervical spine, and then they transfer load to passive structures and deep neck muscles. And that would actually be in keeping with Punjabi's model, where you transfer, if the active system can't do its job, you're going to transfer to the passive system sooner. But it's really intriguing if you start thinking about fatigue as it's not so much a model for neck pain, but it's a model for what altered input from the neck does to the way that the central nervous system actually controls neck function. So I start with that because that's the closest I get to biomechanics, and I thought it was something that your group might find interesting, and because it led to a lot of the other work that my group is doing now. This next study is looking at what neck muscle fatigue, how it affects upper limb proprioception. So we know I'm going to talk about joint position sense in this part of the talk. So JPS is awareness of joint angles in three-dimensional space. And we know that the central nervous system uses the position of the head and neck to interpret upper limb joint position sense. There's numerous receptors in your neck muscles that are responsible for both your central and your reflex connections to vestibular, visual, postural control systems. And if you have altered input from your neck muscles, it's going to affect the sensory input to the CNS and could conceivably then impair the accuracy of your limb movement in 3D space. Interestingly, some of my past work with people who have recurrent neck pain, we found that that group has worse joint position sense accuracy. We know from past work by Knox and Hodges that elbow joint position sense accuracy in healthy individuals actually decreases when the position of the head or neck is passively changed. We know if you fatigue a muscle around a joint, so for instance, if you fatigue biceps, it's going to affect elbow joint proprioception, but nobody's kind of looked further afield and said, if you fatigue the neck, what will that do to upper limb joint position sense? So in this particular study, we wanted to look at whether fatiguing the neck muscles, again, using that 70% MBC, how it would affect elbow joint position sense. Just because of our setup, everybody was right-handed, and because we know neck pain can affect joint position sense, everybody in the group had to be free of neck, shoulder, or elbow pain for at least 3 months before being in this study. So again, the way we measured elbow joint position sense, first of all, we were recording from the cervical extensor muscles so that we could measure fatigue about 2 centimeters away from adjacent to the C4-5 spinous processes. Again, same setup using the 3D investigator system to actually look at, to measure where the elbow was and where it was relative to the head as well. And so the way that we did this protocol is the participants had their eyes closed and the experimenter would actually passively move their elbow to a certain position, hold it, then the experimenter would take their elbow back down to neutral, and that was to avoid changes in the thixotropic properties of muscles, which can actually alter joint position sense. So you always have to make sure that you're not starting from a contracted position. And we kept the joint angle movements fairly small because we wanted to really focus on muscle spindle. We didn't want to skin stretch and so on to come in. The reason I'm laughing as I look back at this, because now we have 3 camera systems, so we're able to measure without having this sling set up. But this was because at the time we only had 1 camera, so we had to have the arm out so that our camera could pick up the markers. So the shoulder joint would have got fatigued, so the shoulder joint was supported. It's like going back in time, really. But that's why we have that set up. That is, however, New Zealand sheepskin. That was one of my early experiments that I brought with me because it made it very comfortable. Okay, so what would happen is we would move the arm into that position, hold, and then the participant had to reproduce that angle, and we looked at the change in the accuracy. So again, this was a slightly different fatigue protocol where they, instead of pushing, they were pulling against a force transducer, and then we looked for, again, when they got a decline in their ability to create that force, and they could not get back up to that 70% level. And same as in the first experiment, significant increases in root mean square and decreases in mean power frequency. So I'm going to take you through, I'll try and go a little bit more slowly on this. Prior to fatigue, we measured absolute error of joint position sense, which was around 4 degrees. The constant error refers to direction. Was there directional specificity? Were they making an error more in deflection or more into extension? And then we had a 4-minute control period. So each person acted as their own control. This was really important because when you do nothing, what you find out is their joint position sense actually improved. And I don't think it's because of it's a learning effect. I think it's because we don't usually pay that much attention to, hey, if I had to reproduce where my elbow is, can I do it? But this is really important because you need to know what happens when you do nothing, but you recreate your measurement, because then it shows when you get these changes following neck fatigue, it's not just because it was going to increase anyway. If anything, it should have continued to decrease because they were practicing. But what we found was that the error of their ability to recreate joint position sense increased significantly. And the other interesting thing is that there was no directional specificity to the error. They were just generally worse at telling where their elbow was when their neck was fatigued. So we know from the first study that neck fatigue affects motor control. Now we know the cerebellum is an important site for integrating sensory input from the neck and spine. It's a key area for motor learning. So this is going to lead to the next two studies, which are going to talk about what is the impact of neck muscle fatigue on the pathways between the cerebellum and the primary motor cortex, and also somatosensory cortex processing in response to learning a novel upper limb task, motor learning task. So again, back to the neck muscles and muscle spindles. We've already mentioned some of the effects of fatigue. And it's interesting, you know, the neck biomechanically connects to the upper limb, but neurophysiologically, the central nervous system actually uses the position of your neck and head to compute where your upper limb is. And so and that passed work by Knox and Hodges that showed that if you alter neck sensory input, it's going to alter upper limb position sense, I think is very important. The cerebellum mediates control of reflexive and voluntary eye and body movements. It adapts the position and movements of your body to execute motor commands. It maintains the representations of your body schema or your body map with respect to changing visual experience. And that's really important, we believe. It's also critical for creating your internal models. So whenever you go to do an accurate movement, it relies on you knowing where is my body in 3D space. So I'm reaching, or any movement I do is with respect to that body schema or that body map. So if the body map is not accurate, how can you have accurate movement that is based on where that map is? And it's very important also for movement coordination. And critically, the cerebellum is really important for motor learning. And in the later stages of motor learning, once we're going from early adaptation to later, you actually see decreased cerebellar activity because you start making the connections for the new learning. You get inhibitory pathways coming in so that you've kind of sculpted the pathway so that the next time you do it, it's much easier because you're beginning to learn. So when we talk about that body schema, it's this postural model of your body. It's very critical for accurate and coordinated movement, but even small changes to that internal model can alter motor skill and limb mechanics. And reaching to objects is a very sophisticated pattern. I won't scare you too much with the cerebellum, but we know that the motor input arrives from your spinal cord, the cortex and vestibular system via your mossy fibers. And so the mossy fibers synapse onto granular interneurons. And then the movement feedback from your muscles and joints arrives via climbing fibers, and that they synapse directly onto the Purkinje cells. And the Purkinje cells learn because from an error signal. But whenever you get an unexpected sensory feedback, that error signal is what causes you to learn. But if you have a situation where the input from your neck is not correct, one of the things that we think might be happening is that Purkinje cell keeps firing because it can't learn because it's not getting the correct information to correct the error. And that's going to probably take me to the end of my career for sure. And I'm sure some of my grad students who will be academics, hopefully in the future, well beyond that to begin to even answer some of those questions. So the next two studies that I'm going to tell you about are more neurophysiological. And the first study involves looking at cerebellar inhibition using transcranial magnetic stimulation, and the second one looking somatosensory evoked potential, so looking at these neurophysiological changes, but also motor learning measured by changes in motor performance retention. So for both of the next two experiments, we had 12 participants for each group based on our power calculations. We had 12 that had their cervical extensor muscles fatigued and 12 that were not fatigued. Again, because we know neck pain can affect all this, we had to use the Neck Disability Index. I often think that in the COVID time, even, well, now we are allowed back on campus for research, but because so many people were at home working on laptops, it would have been hard to find people without neck pain, but back then we could. We, again, were checking the Edinburgh handedness inventory because we were using right-handed individuals just because the task was hard to switch. And the TMS safety checklist was also employed for two reasons. One, to make sure it was safe to do TMS in the TMS experiment, but also to make sure that people weren't taking any kind of drugs or stimulants or anything that might actually affect their neural connections. Okay, and our neck fatigue protocol, again, we did the maximum voluntary contraction pulling against a force transducer, but this time we created fatigue a little bit differently. We had the participants lying prone on a bench, and they actually had to support a 2-kilogram weight until they couldn't do it anymore. And fatigue was defined as an inability to maintain that neutral head posture. And then the rest condition was the participant lay in exactly the same position with no weight suspended. And part of the reason for this was we found with our other protocols used in the earlier experiments, there was a huge difference in how long it took people to fatigue. Some people took 5 minutes, some people seemed to take 20 minutes, whereas this protocol, most people were fatiguing in 5 to 8 minutes. So it's probably, I suppose, convenience is a good reason, but also we wanted to make sure it's hard enough to differentiate, are they stopping because they're bored, or are they stopping because physiologically they can no longer maintain that contraction. Okay, so before we did, in both experiments, before we did our neurophysiological measures, we took our neurophysiological measures, either the TMS measures or the SEPS measures. Then we had the fatigue group undergo fatigue, the control group, as we said, just lay prone. Then we did a motor acquisition task. And then we did our post neurophysiological measures. And then the next day we had them come back to look at retention. And we did all the normal things, but we used a mixed design repeated measure analysis of variance to compare the change with fatigue, but also to compare the group effect. Okay, so the first one is the TMS one. I'm going to try and go slowly so that you can understand a bit about TMS if you don't know much about it. I'll talk a little bit more about motor learning as well. So we know acquisition or improvement of a motor skill with practice is the definition of it. We know that practice induces plasticity of both your motor cortex and your cerebellum, and that you get cerebellar plasticity. And we know from some of our own groups' past work with neck pain participants that neck pain can impact motor learning, but it also affects SEPs related to the cerebellum and motor cortex. No one's really looked at fatigue. And from my perspective, looking at fatigue was really important for two reasons. One is because fatigue is becoming ubiquitous. Interaction with technology. People always have their head bent down. Or in different workplaces where the head's up. There's all kinds of reasons why fatigue is more prevalent. But as a basic scientist, what I loved about fatigue is in the lab, you can create something where you're changing sensory input, and then you can really say, this happened because the neck was fatigued. When you're dealing with the clinical population, there are so many other things that can affect it. Psychological, they might not have just neck pain, they might have back pain and shoulder pain. So it's kind of nice. It's the scientist in me likes the idea of something that you can control a bit better. Okay, so the way that this motor acquisition task worked, let's see if I can get the animation to play. Of course, it probably won't unless I can. Let me just escape. Oh, there we go. Okay. So what happened is the trace came down the screen and they had to track. And the green means they were doing well. The yellow means they sort of missed a little bit. And these sinusoidal waveforms, they varied in frequency and amplitude. We had 4 different waveforms. The easiest everybody could do, the hardest one was very challenging, and the other 2 were in between. And that was to allow for continuous learning, and so also that everyone was able to learn. Okay. And as we mentioned, the color coding gave them feedback as to whether they were accurate or not. And that was using Leap Motion software. And we spent a lot of time actually validating the task. One of my other students, Daniel Andrew, who was a master's student at the time, looked at the task itself and showed that it did lead to continuous learning. And the way we measured the error was literally the difference between the original trace and how close they were in their attempted trace. So we hypothesized, of course, that neck fatigue would lead to less cerebellar disinhibition following novel motor acquisition. So let me explain that a little bit. When you acquire something new, your cerebellum has to temporarily, we call it disinhibit, because when you learn something new, the cerebellum sculpts these pathways through inhibition. So a great example I sometimes use is when I went to New Zealand and, I knew how to play softball, but of course they wanted to try and teach me to play cricket because that's their sport. And I would start with my cricket bat down because that's what you're supposed to do. And next thing I would be holding it like a baseball bat. And then all these New Zealanders would be rolling around on the ground in hysterics as if this was the funniest thing ever. But the point was that I had a motor pattern already in there to hold a softball bat. So if you put a wooden bat in my hand, I would keep reverting to that pattern. And to learn something new, you've got to be able to let go of that, disinhibit to lay down the new pattern. So if you can't disinhibit, it's hard to learn something new. So we thought that neck fatigue would not only lead to less cerebellar disinhibition, it would lead to worse motor learning. So transcranial magnetic stimulation is a safe way to stimulate. You can stimulate both over the motor cortex. I want to make it clear in my lab, I am only using TMS, regular TMS, to study excitability of motor pathways. There is also something called RTMS, or repetitive TMS, that some researchers use to temporarily either increase or decrease excitability of different cortical areas, but that's not what I do in my lab. I would just study motor pathways. So this figure of 8 coil produces a very focal magnetic field, which leads to neuronal flow in axons, and then you get transsynaptic excitation of neurons in the motor cortex. And then this is the double cone coil over the cerebellum. It has much deeper penetration because the cerebellum, it's sort of a deeper structure to activate. Now the cerebellum has no direct output. But it does have a pathway between the cerebellum, and the ipsilateral cerebellum connects to the contralateral motor cortex. And as you may remember, the contralateral motor cortex, so your left M1 is what activates your right hand muscles. And but the right cerebellum connects to the left M1. And so what you want to do ordinarily when you're learning something new is those cerebellar pathways should be disinhibiting. So I have a tool now that allows me to study those pathways. And so what you do when you stimulate here over the motor cortex, you get something called the motor evoked potential, or a MAP. You record it with EMG electrodes over whatever muscle is your target muscle, because you can move this figure of 8 coil around until you can activate just the muscle that you are interested in. And so here's our figure of 8 coil. Here's our double cone coil over the cerebellum. And there's our recording electrodes, in this case over the first dorsal interosseous. Now, if you stimulate the cerebellum 5 milliseconds, 5 to 8, but we went with 5 milliseconds in advance of M1, you actually can inhibit the output. So if the red was your normal motor evoked potential, if I stimulate the cerebellum first, I decrease the amplitude of that potential. So what we wanted to do is look at cerebellar inhibition. We looked at it prior to fatigue or rest, the rest control condition. Then they did acquisition. Then we looked at cerebellar inhibition again. And then the next day they came back and we looked at retention. So the first thing we found when we looked at their accuracy on the task, here's the control group. So this is proportional to trace error. So getting lower is better. It means there's less error. And what you see is a classic learning curve here where pre to post, so acquisition, they've improved And at retention, they continue to improve. When you look at the group that had the fatigue neck muscles, what's really interesting is not only did they learn, they had less improvement at acquisition, but at retention, it's like they plateaued. The slope of their continued improvement was much less. So there was a significant interaction both post-acquisition and at retention between the neck muscle fatigue group and the control group. Oop, went too fast there. Okay, I'm going to try and explain this really carefully. So ordinarily, 100%, so all of this is relative to your 100%. If I was just to stimulate M1, I'd see 100% MEP. So what you're seeing, pre is the dark lines. So let's just focus on the control group first. When you look here, here's your 50% inhibition. So I stimulate, I find the level of the stimulator that inhibits that motor output by 50%, and then I test 5% above that, 10%. I'm averaging 10 to 12 sweeps at each of these points. And so what you get is this nice curve that shows inhibition, just stimulating the cerebellum. We haven't even done anything at this point, right? And remember, I mentioned before that when you learn a new motor skill, your cerebellum needs to disinhibit. So what happens here then is when we look at this after they've done the motor acquisition, this post-intervention curve, there is significantly less inhibition from the cerebellum to the motor cortex. And that is because the cerebellum has disinhibited in order for those people to actually learn that new skill. Now, in the fatigue group, it's actually quite frightening because they did not disinhibit at all. So they did the motor learning task after, but their neck was already fatigued when they were learning, and they had no disinhibition. So now we've got a situation where we can show that both neurophysiologically and behaviorally, fatiguing your neck affects your ability to learn a new motor skill. We know that TMS over the cerebellum influences discharge from extracerebellar projections to cerebellar nuclei via your mossy and climbing fibers, likely due to GABAergic neuron inputs from Purkinje cells. We think again that the sensory neurons that project to the CNS as a result of the changes in motor behavior, that creates your error signal that weakens your Purkinje cell outputs, and that's what you need to enable motor learning. And practice leads to increased cerebellar cortex activity at the early and late stages of adaptation. Neck fatigue alters this. And like I mentioned earlier, what my lab thinks is it's because the Purkinje cells are caught in this loop where they're getting a continuous mismatch between the motor command and sensory feedback. Okay, so last study. Sorry, I know that that's a lot of complex neuroscience if you're not used to it. I was very proud of Mabube for this. So one of her more recently published PhD studies was selected for the cover of Brain Sciences, which is kind of a nice accolade for her. It's a nice way to end your PhD, put it that well. Actually, she was a postdoc by then, but very, very nice. So that's what I'm going to talk about in this study, is looking at somatosensory evoked potentials. So the way you record somatosensory evoked potentials, it's a form of EEG or electroencephalography, but you stimulate a peripheral nerve and you record. Now we use caps in my lab, but at this time we used electrodes that were applied to certain sites over the scalp using the International 1020 system. And we also recorded peaks, spinal peaks reflecting dorsal horn and so on, that were recorded over the spinous process, and the stimulating electrodes were over the median nerve of the wrist. We actually stimulated at 2 different rates because we did some past work that shows there's one of the peaks we were really interested in, the N24, which is the cerebellum to somatosensory cortex pathway. And it kind of rides on this N30. And if you stimulate a bit faster, the N30 attenuates. So that's why we stimulated at 2 different rates and averaged 1,000 sweeps for each somatosensory evoked potential. So when you measure these potentials, you're measuring the peak to peak amplitude of the potentials. And so 24 means a peak that occurs roughly 24 milliseconds from the point of stimulation. And short latency SEPs occur for the upper limb, 8 to 30 milliseconds, and medium latency is sort of the 60 to 100 millisecond period. These are peaks that are not yet cognitive. This is pre-cognitive processing. So what it's saying is, if you get differences in these peaks, it's saying that the information arriving at the cluster of neurons that generates the activity of those peaks is fundamentally different in a different condition. And you don't even know it. So you can't fix it because it's not even yet at the stage of consciousness. So very early on in neural processing, it lets you look at how these altered sensory inputs are affecting neural processing in the brain. So just to give you some ideas here of the different loops, and I'm not going to go into exhaustive detail about all of them, but some of the peaks, like the N9 is recorded over your brachial plexus. That's just, we don't want that to change. That's the one that shows us that we have a nice stable incoming volley to the central nervous system. N11 is the arrival in the dorsal horn. And then the N20 is the primary sensory cortex. N30 is, we call it the sensorimotor integration loop, but it reflects activity in basal ganglia, supplementary motor area. So the point being that we do have ways to look at certain clusters of neurons and how they're affected. So we use the same task, same setup. The only difference is instead of TMS, we were using somatosensory evoked potentials. And again, same thing where we showed that we got significant fatigue happening, decrease in mean power frequency, and an increase in RMS. And again, as you might expect, similar changes because the task was similar, where the fatigue group had greater error. They did not decrease their error acquisition, although interestingly, this time they did do a bit better at retention. But fascinating to us anyway, the two peaks where there was a significant difference, significant interaction effect between the fatigue group and the control group were the N24 set peak and the N30 set peak. And what this is telling us, again, the N24 represents the pathway from the cerebellum to your sensory cortex. As I mentioned, the N30 represents activity in this complex sort of cortical subcortical loop between basal ganglia, thalamus, premotor area, primary motor cortex. The way they work out the neural generators of these peaks, in the early days, some of it was direct recording during neurosurgery, some of it was lesion studies, but more recently, there's really amazing mathematical software available where you can actually do source localization, and you can actually, the software is literally resolving the vectors to say, here's the source of neural activity. And the N30 is interesting because it actually has these multiple neural generators. So you can really start drilling down now and finding out where the changes in sensory process are happening. And so we know then that the cerebellum is affected by neck muscle fatigue, as well as your sensory motor integration pathways, and that worse performance accuracy indicates that input from the neck impacts upper limb movement performance. So thank you to all of the students who did the fantastic work in these studies and some of the studies that went before them. And thank you. This is just a fun picture of the lab from back when we could still get together in person. So thank you.
Great. Bernadette, thank you very much. You're welcome, I think, to turn on your video if you like. I think that will work for you.
Oh, let's see if it'll let me. Okay, I'll escape from here.
There you go.
Oh, okay, great. So now it works. Nice. Okay.
We have that fixed up. So that's good. From an audience perspective, please submit your questions through the Q&A. And that's how we'll handle them. And I'll read the questions and then out loud, and then Bernadette can answer them, please, for the next few minutes. That was great. That was very interesting. Let me just start with a couple of questions here. I was really intrigued by the fatigue protocol that you used when you were the person was lying on their tummy there, and they're trying to lift the 2 kilogram weight. You said fatigue takes place in about 5 to 8 minutes for most of them, I guess, huh? And yeah. How do you discern when when they are fatigued? They just can't.
Yeah, literally, we used an inclinometer, and we literally, if they could not maintain their head in the neutral position, then that was defined as fatigue. Like if they couldn't, we would try and encourage them, come on, get your head back up. And, if they can't do it, we're continuing to encourage them, and they just cannot maintain that, the neutral position, then that was the performance measure of fatigue. So the electrophysiological measures were just to confirm that physiologically they were indeed fatigued.
Right. And I guess I'm curious about the fiber composition of these neck muscles. What are they?
They're mainly type 1, which makes sense if you think of it, because you've got to hold your head up all day.
Right.
But there are some studies that have actually done biopsies in people with chronic neck pain, for instance, that show that there's actually, they actually have a lower proportion of type 1 fibers. So again, it's interesting again as to what might be happening neurally because of the, and that's in the case of pain, but this was just in fatigue.
Right. When you say mostly type 1, are you talking like 70-80%, or?
Yes, 70-80%. Yeah.
Oh, okay. Yeah. Okay, so they take a while to fatigue. If they were type 2 fibers, they would fatigue probably a little bit faster. But anyway, do you suspect there's any fatigue, any sort of fiber type related difference in how these signals would go to the cerebellum to inhibit learning? Or
that's a really interesting question, because when you think of it, I mean, as far as I know, I don't think the muscle spindles are any different in terms of signaling, changes in length. But when you think of the fact that the fast twitch fibers have to respond more quickly, that would be really, really interesting to see if there are any differences in projections. Now I'm going to go and look and see if there's any animal work that's done retrograde labeling to do that.
Yeah. Okay, let me go to the Q&A. Let's see. Thanks for your talk, says Matt Russell. Thanks for your talk, Dr. Murphy. Enjoyed your anecdotes. I was curious how you plan to isolate your Purkinje cell hypothesis.
Well, the thing is, I couldn't do it in people for sure. I'd have to look and see what's been done in the animal work. But there are things, I mean, we do know that the Purkinje cells, that is the error signal. And if the error is not corrected, the Purkinje cells will continue to fire. So certainly that is a known fact, but what we don't know if indeed that is the reason for this. Funnily enough, I did a guest lecture for first years in first semester, and one of the questions the lecturer asked me is, if somebody gave you $100,000 to do whatever study you want tomorrow, what would you do? And I said, actually, I would collaborate with an expert in cerebellar MRI, and because it's really tricky to do cerebellar MRI, and I would redo some of these experiments in the scanner, because then you could really localize where it's happening. The neurophysiology stuff is better for timing because you can get really great temporal accuracy, but the MRI gives you the spatial accuracy. But I didn't think about the fiber types. That would be really fun.
Yeah, okay, yeah. Dr. Janessa Drake says, thank you very much for such a wonderful talk. Can you share your comments on whether folks that routinely exercise have less errors in joint proprioception in the fatigued condition?
Oh, that's really, really interesting. I suspect in regular joints, that would be true, like as in if you fatigue biceps and look at your elbow joint. One fascinating thing that's going on right now is I actually have a study going right now with people who have recurrent neck pain. And there's a lot of really intense exercises. There's a few, in the past, I found CrossFit athletes, they're the worst. They get so many crazy injuries. So I would say it's a U-shaped curve. I think the moderate exercisers quite possibly are more fatigue resistant. I also think technique comes into it, because a lot of people, they kind of overdo their exercise, or they want to outdo the person next to them, right, in the gym. So they're really going hard, and then they're using their neck muscles for things that they shouldn't even be activating their neck muscles, but they are, because they're lifting weights that are too heavy and their traps come up, and or they're doing some kind of abdominal work, and they've got they're so a lot of it I think comes down to technique. So I don't think it's unfortunately, it's not always a panacea, because some of the worst necks I'm seeing are from the most intense exercises in this study. And it's not even an exercise study. It's just because it's a student population, you've got, like every kin program the world over, a lot of really intense, hardcore, crazy exercise people.
Yeah, that's right. Yeah. Dr. Arthur Cheng, thank you, Bernadette, for your talk. Have you assessed any contractile properties in the neck, in the muscles of the neck pain patients? Just wondered if neck fatigue is explained by any muscle-related factors, or if it's all supraspinal fatigue related.
Well, for sure, I think in the neck pain, I haven't done it, but I did cite a study on one of my slides where they did show that there was a change in fiber type in people who had chronic neck pain. So I think in that situation, it may start initially kind of, and I don't know, it's interesting with this recurrent group that I studied, whether they've already progressed to the biomechanical changes or whether it's all, but certainly with the fatigue, it's interesting because I am presuming, I don't know, but I'm presuming it's mainly central fatigue that I'm dealing with. It may progress. Actually, I shouldn't say that. That's incorrect, because it does progress. The peripheral, of course, over time, you're going to get the peripheral changes as well, because this is a funny story too. Talk about getting embarrassed during a talk. So I was doing a talk at McMaster a few years ago, and I got asked if doing the fatigue protocol in a healthy person gave them neck pain. And I said, oh no, I don't think so. You know, because they never say anything on the day. And one of the students in the audience was an Ontario Tech student who was a master's student doing her PhD at Mac. She's going, but I had DOMS for 3 days. And I'm like, thanks for that. So I think the answer is that the fatigue probably does, like the protocol probably does create both central and peripheral changes. And it may be that fatigue is actually quite a good model for what happens over time with neck pain, but I don't think it would be terribly ethical to be fatiguing people to try and create irreversible chronic neck pain.
Yeah, no. I would imagine there's some local acidosis though in that neck muscle that is not, I would think so, right?
Absolutely. And one of the other problems, if somebody has neck pain, there is quite a lot of spasm, and it's very interesting to think, of course, you're going to maintain arterial perfusion, but the question is, does that tight muscle, do you get some changes in the capillary beds, right? And is it underperfused relatively? And they're way back when I was doing my master's, I was interested in sort of repetitive overuse type things. And, even 5% of MVC repetitive contractions will affect blood flow if you're doing it for long enough, right?
That's
So
true.
I think that's a really important factor. And we often forget about that.
Yeah, no, that's true. So the chair of the student committee, Daniel Deira, is asking a question. He and his group are the ones that invited you to speak today, Bernadette. Daniel says, thank you for your talk, Dr. Murphy. Does time to recover from fatigue differ between neck pain and healthy subjects?
I think that's actually a great question. And unfortunately, I've only done 2 studies comparing the 2 groups. Like, I don't usually fatigue a neck pain patient. The study I showed you at the beginning today was because we were really interested in testing Punjabi's model about that active passive subsystem and the transfer of load. So that's why we kind of did that work. But I don't know, and it'll be interesting. I could ask Mabube if anecdotally she found any differences. But a study I didn't tell you about today, we looked at shoulder movement in people with neck pain versus healthy controls and the effect of fatigue. Fatigue made the healthy controls look like neck pain. They move their shoulder abduction, instead of being straight out, they come more forward. And so it changed their motor control. So it'd be really, really interesting, I think, to look and see if they take longer to recover, because if there is that fiber type change, that would make sense.
Yeah, okay. Dr. Janessa Drake, could you share your thoughts on how aging plays into the findings of neck fatigue and cerebellar disinhibition needed to learn the motor acquisition?
Wow, great question. So one thing I should have mentioned, of course, like so many university researchers, a lot of these were students. So they were in the 18 to 30. And funnily enough, as a neuroscientist, I purposely wanted 18 to 30 because there is a higher degree of neuroplasticity. So certainly when you're older, you can still learn. I just managed to learn
Thank goodness for that.
Yes, I managed to learn the odd new motor skill myself even. But it does take longer. It is harder. So definitely there's more plasticity when people are young. But it's a great point because if you have aging decreasing your ability to learn and you compound it with neck pain, you could literally be in double trouble. So that is an excellent question.
Right. Another from Arthur Cheng. What's the best way to train or rehab the neck muscles to mitigate neck fatigue?
Interesting. Okay, I think motor control and cueing is really, really important. One of the things that I do with patients, I never give them exercises on their first visit, because if you have maladaptive motor control and you try and exercise when you're already in a maladaptive state, you actually exacerbate your maladaptive muscle recruitment pattern. So the only kind of exercise I give people initially is activation exercises. And I'm sure you guys are familiar with, everybody knows, or most people seem to know about the transversus abdominis and activating your transversus abdominis when you're doing any kind of lifting. But it's the same with the neck. Your deep neck flexors, your longus colli, longus capitis, they're the ones that are most commonly injured in whiplash injuries. But any kind of stuff whiplash-like injuries. So what you need to do is reestablish them activating, because what happens instead is things like sternocleidomastoid ends up, it's supposed to be a prime mover and it ends up having to be a stabilizer because your stabilizers are switched off or inhibited. So I think a really important thing with rehab is first activate. One really easy exercise is if you press your tongue against the roof of your mouth and just do really controlled neck flexions, that will help activate your longus colli. So that's one way to do it. And the other really important thing is if you're doing any upper limb exercise, lifting weights, make sure you're not using your neck to do it. Because I think that's another real problem people get into is they're not isolating upper limb and they're exacerbating the neck muscle problem.
Right. I'm glad you brought up whiplash, because we have folks here in the Muscle Health Research Center that study whiplash. And so that being a neck injury, I guess, is it similar mechanisms involved in fatigue influencing the learning as well as whiplash induced injury? Would mechanisms be the same in terms of that, or?
Well, given that we do know that with neck pain, it influences motor learning, I think it would be highly likely. Like I've published a lot of work on neck pain showing really similar findings to the fatigue, but I just thought this group might be more interested in the fatigue story.
Sure.
But yeah, the difference, of course, with whiplash is it's a spectrum, right? You've got grade 1, 2, 3, and some people have, I think, fairly minimal injury, but the CNS is responding like it's a major injury, right? So it's got those muscles in spasm. And what you've really got to do is re-educate. But there are some people, of course, who have a very severe kind of whiplash where they literally have tearing, even to the point of being unstable, in which case you want all the muscle spasm you can get because it's actually truly is protecting you. So I think that's one of the challenges with whiplash. It's not one size fits all. Yeah.
Right. Okay. Michaela Slavin asks, interesting research on that note. Is there work that sheds light on the effect of stretching the neck carefully and yoga on sensory motor integration and learning?
Well, the first answer is I don't know, because I haven't followed that literature. It wouldn't surprise me if it did, in the sense of there is some good literature on this sort of activation training of the deep neck flexor muscles. And I know that a lot of different types of yoga, or certain types of yoga, focus very much on that activation. The thing you have to be careful with stretching, especially if you've got either a fatigue neck, because we already know there's this mismatch and possibly inaccuracy in signaling. In the muscle spindles change their firing pattern when you're fatigued. So they may not be accurately signaling your muscle length. But the other thing that happens, of course, is your Golgi tendon organs come in and switch things off. So if you can actually overstretch quite easily. So you have to be really careful. That's where I think the activation comes in. The other interesting thing is, you know, I'm trying to think of a good way to explain this. I'm thinking back to New Zealand here. So when you had sheep shearers that are always in a forward flex posture, I saw some fascinating research when I was there, and they are always forward flexed. So when you ask a sheep shearer, so they're always down like this doing their work, when you ask them to extend their back, instead of doing this, they extend their neck. That's all they can do. They have no lumbar extension left because they're habitually forward flexed, right? So that's an example of where you've overstretched, right? And now your connective tissue has major creep from which it's not recovering. So I think everything is a balancing act, but I certainly think yoga has a lot of potential as long as the cues and the instructions are correct.
Yeah, yeah. I guess maybe one final question. Is there a patient population with cerebellar defects that would be, if you could tap into those folks, would they be helpful in sort of identifying some of the pathways that you're interested in? Or
well, interestingly, that is how we know about the pathways. There are such people, and that's not with this particular task, but I've done other throwing tasks, and that was related directly to studies on cerebellar patients because they have, of course, terrible accuracy. So the neck pain patients aren't as bad as the cerebellar patients, but they're sort of bad enough. So we definitely have, I think the fact that two different studies using two totally different neurophysiological techniques both point to alterations in cerebellar connections. It's pretty clear to me that the cerebellum's involved, but to answer exactly how, I would have to do some fMRI work.
Yeah, sure.
So I'll have to come to York. So if anybody wants to collaborate,
yeah,
we can do it.
That's a nice invitation. I'm sure people listening might want to jump on that. I guess I have one more question from Matt Russell. We'll end it here because we've given you a lot of questions. With the quicker transfer from active to passive support systems, do subclinical neck pain patients show a higher proportion of neck tissue injury?
Well, this is a thing, like, we don't know, but I think that's what I'm trying to get at, is that could be why they progress from being recurrent or subclinical to chronic. Because they are, like, I guess my point, again, as a scientist is, sure, you could say, okay, well, they didn't get treated soon enough. That's all true. And they just let their pain get worse. But my question is, how do these neural alterations, how could they increase the injury risk and actually lead to the progression? Because I think if we can show that, then we have a lot more impetus to intervene with exercise, with whatever it is, to actually stop this recurrent stuff from becoming chronic.
Right. Very good. Very good. Well, everyone, I think we should thank Bernadette very much for her very, very nice presentation. That'll be the end of our seminar for today. Bernadette, please stay with me for a moment. Thanks for coming, everyone. See you next time at our next seminar in a couple of weeks. All right.
Thanks, everyone.